My name is Drew McKellar. I'm a graduate student in the COPPA lab at Seattle Biomedical Research Institute, where we research infectious diseases, particularly those that are more prevalent in developing nations and aren't readily addressed by a lot of research programs in the U.S. And here we work on the liver stage of malaria infection. It's a little-known area of research, especially even in the malaria field, which is a small enough community in the life sciences. Malaria is a disease caused by the parasite Plasmodium. So the infectious cycle begins when an infected mosquito bites a person, and then they start to migrate around under the skin until they reach a blood vessel and are transported in the blood to the liver. And in there, the parasite changes its form, undergoes very rapid and dynamic growth, and forms tens of thousands of daughter cells, which eventually get out of the hepatocyte and go back into the blood circulation. This is when the disease proper starts, with the onset of the symptoms of malaria. The life cycle of the malaria parasites is very complicated. It requires a lot of different genes and a lot of genetic organization and complexity on the part of the parasite in order to achieve this. The genetic program that the parasite follows to get through its life cycle is very intricate, and we're interested in studying that particularly. Following World War II, with the advent of chloroquine and some other new drugs, and DDT as a very powerful insecticide, there was a pretty strong campaign to eradicate malaria. It was believed that we had all the tools, we had enough understanding of the parasite, the life cycle, the vector, and the right drugs to finally deal with this. So as the parasites continue to evolve resistance against the drugs that we use to clear them from the bloodstream, and as the mosquitoes can evolve resistance to the insecticides that we can use to kill them, it becomes a constant evolutionary arms race between the human host and what we can do to stop the parasite, and the parasite, which has to survive and has done well at that role for millennia. What you should know is that there are 4 different species that cause the disease in humans. Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale. Of those, the worst one is falciparum in terms of the number of deaths that it causes. And there's also additional concern about Plasmodium vivax, simply because while it doesn't cause as many deaths, it's kind of has a stealth mode in that rather than completing the life cycle upon infection, it can stop in the liver and lie dormant and then reemerge years later, so that an infected person or someone who travels to, say, Africa, contracts the disease, returns home and becomes treated, but within that time frame they can become bitten by a mosquito that, the mosquitoes that can carry malaria do still live in the US, and they can get a local infected vector. Malaria does continue to pop up, primarily through travel of Americans and Europeans to infected regions. And if we continue to ignore it to the extent that we had in the latter half of the 20th century, I think it would just be a matter of time until it returned. But there is enough motivation at present and enough people interested in the problem that I think it's more likely that in the future we will be pushing malaria back rather than having it reinvade the US. There are several different things that we can do to stop malaria. The principal thing that is done is when a person becomes infected, you go to your doctor and get prescribed a drug. These drugs are sometimes expensive, sometimes have bad side effects, and aren't always in immediate supply. But in any case, once you get that drug, you take it for a certain course, and oftentimes they can be very effective. They can actually clear the parasites. The problem is, pretty much any drug you can come up with, the parasite has a chance to evolve resistance to it, and then the drug becomes ineffective. But the situation gets more and more complicated as this dynamic continues because researching new drugs is very expensive, and a lot of times the newer drugs that we come up with are even more expensive than the ones previously available or have worse side effects. Drug design and development and production and making it available to the infected people, that's a big issue. But that is the main way that we deal with malaria nowadays. The other crucial thing that is done at present is to deal with the vector. So you can treat with drugs, try to kill the parasite once you become infected. You can try not to become infected by controlling the mosquitoes, or at least, if you can't kill them all, keep them from biting you. And the third thing that's potentially feasible but hasn't emerged yet as an actual tool is a vaccine. And the way that that's being done is all just conceptual right now. We know that vaccines can work in mammals and even in humans against malaria, but so far we haven't found a way to package it and produce it so that it's truly accessible and that we can deploy it as a wide, safe tool for use in endemic regions. So vaccines against malaria can be directed against any of the three major life cycle stages. You can try to treat blood stages in humans. You can try to treat it in the liver. And additionally, although mosquitoes don't have an adaptive immune system, we can direct vaccines against mosquito stages. So how that works is, in the case of the mosquitoes, the sexual forms of the parasite that emerge in the mosquito gut and go on to infect the rest of the mosquito aren't normally seen by the human host. So we don't direct any antibodies or cells against those stages. But if we are exposed to those forms in a vaccine, so that you purposely inject someone with some antigen, some small part, some molecule of the sexual stages, they can produce antibodies against them, which could block the sexual fusion and continuation of the mosquito infection. People have shown in certain models that this can be done so that when a mosquito takes a blood meal and the parasites come up in the blood and are ready to infect the mosquito, the antibodies circulating in the mammalian host can stop the parasite from infecting the rest of the mosquito. The idea being that's called a transmission blocking vaccine. If you can stop just mosquitoes from getting infected, they can't infect new people. And therefore you've broken the cycle of transmission. Any existing infections can hopefully be dealt with by drugs. You just don't have to worry about people becoming infected again. And the third thing that we can do and that we're interested in our lab is a vaccine against the liver stage of malaria infection. And that is because when a mosquito bites the person, very few parasites are introduced, and they have to go through the liver before they can get into the blood and go through that cyclical stage where they really amplify the numbers of parasites in the person and where they really cause all the symptoms. So if we can block it at that step, once it gets into the liver, if that is a target that we can wipe out, then a person that is vaccinated could potentially never become infected, never come down with the symptoms. And that is, at current, the only absolute way we know of to use a vaccine to prevent somebody from becoming infected with malaria. The idea with a genetically attenuated parasite, which is what we call it when we just mutate or knock out one gene, is that it's a very well-defined system. We know exactly what's wrong with it. We can specify, we can home in on exactly where it's stopping. We know just what to expect when we put it into a mouse or a person. And we're getting ready to do that in the near future. There are going to be clinical trials conducted here in Seattle at this institute using the genetically attenuated parasites in humans. And someday we hope to actually have maybe a single molecule in a syringe that you could inject into someone with no risk of infection and very few side effects, that would be much cheaper and easier to make and to deploy, and that could actually be a tool in malaria eradication globally. If we can move towards that end, that would be the ideal vaccine against malaria.